Today, I'd like show you my thesis on inhibition of aggregation and dissociation of aggregated and tau proteins by CAC717, the calcium hydrocarbonate. The figures in the left image is the CAC717 by electron microscope, the mesoscopic structures, and versus the light amyloid beta aggregate and tau protein aggregate as immunohistochemistry. Okay, the first I briefly introduced the action of CAC717. CAC717 mesoscopic structure in aqueous solution undergo electron circulation, such as discharge and supplementation of electron. When it touches cell membrane, etc., it emits a pulse wave, terahertz, so 10 to the 12 hertz. The wavelength of the pulse is from 800 nanometer to 100 micrometers, a sub-millimeter wave, which is close to the far infrared. Macromolecules in the body, such as lipid, sugar, protein, etc., can undergo molecular vibration and structure changes induced by terahertz waves. However, the energetic effect of pulse waves are very small in nano-sized, 50 to 500 nanometer microenvironment. Thus, higher molecular organisms such as plants and animals, but not bacteria or virus, do not show harmful effect. But sometimes cell activity may occur depending on the wavelength. So I introduce my experiment and result, the inhibition of aggregation and dissociation of aggregated Aβ and tau protein by CAC717. The experiment conducted at the three levels. The first is molecular levels. Second experiment is cellular levels. The third is in vivo levels using Alzheimer's disease model of Drosophila. The first, the molecular level 1. The material method of the Aβ. Human recombinant Aβ1 to 42, peptide 4349V was solubilized in 0.02% ammonia water. Final concentration is 250 micrometer and stored at minus 80 centigrade. The final concentration was adjusted to 50 micromoles by adding 50 millimole PBS, pH 7.5, and 10 millimole sodium chloride. Aβ142 solution, 600 microliter, was reacted at 37 centigrade for 4 hours to obtain aggregate. To 5 microliter of Aβ aggregate, 5 microliter, so equal volume of undiluted, or 2 times and 4 times the dilution of CAC717. I used the 3 lot for the reproducibility, A, B, C, were added, incubated at room temperature for 30 minutes, and then analyzed by Western blotting with Lemley buffer. In case of the tau protein, After showing 1 microgram per microliter of human recombinant mutant tau, fluorine 301 serine tau aggregate, stress mark Bioscience Inc. On ice, undiluted 2 times 4 times diluted CAC717 5 microliter were added to 5 microliter of aggregate and incubated for 30 minutes at room temperature. A lemon buffer was added and analyzed by Western blot. The results shown in Fig 1. This is Fig 1. The primary antibodies were mouse monoclonal antibodies, anti-A-beta, beta-lactin, and anti-tau. This is a tau 12 Merck monoclonal antibody 2241. The secondary antibodies was horseradish peroxidase conjugated anti-mouse IgG antibodies. Bethel, A1960P. CAC717 dissolved A-beta and tau aggregate as shown in the figure in a concentration-dependent manner. Dissociation appears to be clearer with tau protein. Molecular level 2. A-beta aggregate dissociation by CAC117. The amount of A-beta aggregation is measured by the fluorescence intensity of theophlavin T. Intensity increases with the progress of the aggregation and decreases with A-beta dissociation. On the other hand, the amount of A-beta monomers is measured by ELISA using monoclonal antibody for A-beta. In this case, the more monomers, the stronger the reaction, and when it becomes aggregate as polymers, the epitope is hidden and does not react with monoclonal antibodies. Addition of CAC770 to A-beta aggregate significantly, the probability is 0.05, decrease fluorescence intensity, so theophlavin T as Fig 2, compared to the sample with distilled water added control, CT. Dissociation of A-beta aggregate was confirmed. As aggregate dissociation by CAC717 as time course. The time of A-beta aggregate to dissociate with CAC770 was varied as 5, 15, and 30 minutes. Oligomers and monomers with antigen epitope generated was measured by ELISA. The reaction of CAC770 was stopped by addition of herpes buffer. Results shown in the Fig 3, the time course of dissociation of A-beta aggregate. The dissociation of A-beta aggregate was confirmed after 5 minutes. It is a bit astonished for me. 5 minutes of a reaction. There was no difference in reaction time between 5 and 30 minutes. So the dissociation is occurred rapidly when treated by CAC717. Inhibition, this is the result is the dissociation. This is the inhibition of A-beta aggregation by CAC717. The theophlavin T intensity of aggregation of Aβ was measured as before. When CAC770 was added to Aβ 142 solution, 10 micromole, theoflavin intensity was significantly lower than control distilled water. Inhibition aggregation is significantly greater than dissociation of Aβ aggregation. So figure 4, inhibition of Aβ aggregation. The probability is less than 0.01 as compared to the dissociation of Aβ. And inhibition of Aβ aggregation by CAC717 checked by theoflavin as dose response. So Aβ aggregation was measured by theoflavin B as before. Aβ aggregation was inhibited by addition of CAC717 in a concentration-dependent manner as undiluted 2 times and 4 times and 8 times diluted. And the dose was significant even at 8-fold dilution. So inhibition is relatively more effective than dissociation of Aβ aggregation is clear. The second is the cellular levels. I use the mouse neuroblastoma cells in this case. So inhibition of tau aggregation by CAC770 in cultured neural cells. Mouse neuroblastoma, neuro2A cells, N2A cells, are maintained in the albicus modified Eagles medium plus 5% fetal bovine serum, DMEM plus 5% PBS, and cultured in 1% FBS DMEM to prevent cell overgrowth during experiment. L2A cells were seeded at a density of 3 times 10 to the 4 per square centimeter to 4 wall plate, with 6 wall plate or 12 millimeter cover slip coated with 0.1% polyethylene with film the first day. And then day 2, polyethylene max was used to gene transfer human recombinant mutant tau, P, so proline 301 lysine tau into N2A cells. The next day, on day 3, CAC717 and lysosome inhibitors, so peptidine, E64D, and rifepsin, each adjust to the final concentration of 10 micromole. Abbreviation is a PEL, PEL, were used for lysosome protease inhibitor and treated by total media exchange. On day 4, cells were collected and used for the various experiments. The plasmid expressing the human variant of tau protein, tau form, familiar from temporal lobar dementia on chromosome 17, so distributed by Dr. Akihiko Takashima. This slide is the all results for the immunohistochemistry. Intracellular accumulated tau aggregates are degraded in lysosome via autophagy, the normal stage. To confirm that reduction of the tau by CAC770 is not due to lysosomal degradation, inhibitors of lysosomal protease were added together with CAC717. The results show that even in the presence of the inhibitor, CAC significantly reduced intracellular tau, indicating that the reduction of aggregate tau by CAC717 was direct and not lysosomal-mediated proteases. Immunocytological staining confirmed that CAC717 treatment clearly reduced intracellular tau even in the cells with significant intracellular accumulation of autophagosomes, the lane of the LC3, due to proteolytic enzyme inhibitor, VEC7. The abbreviation is the tau protein P301L. LC3 is the autophagosome accumulation. DAP1 is the cell nucleus staining, merge, superposition, and CAC power. So CAC power is the CAC and a protein in lysosomal degradation enzyme inhibitor, a cathepsin D and serine protease inhibitor. The second is triton soluble and insoluble fraction from B301L tau expression and CAC 717 treated N2A cells were analyzed by Western blot. Each sample was adjusted to 10 microgram protein electrophoresis on the 10% polyacrylamide amide gels and transferred to the PVDV DF membrane, milk. After blocking with 5% skim milk PBS containing 0.1% 2020, the membrane was placed in the hybrid bag with anti-tau antibodies, 5,000 dilution, and anti-beta-actin antibodies as 10,000 dilution, and left overnight at 4 degrees. After washing the membrane with PBST, it was treated with secondary antibodies, 20,000 dilution, at room temperature for 1 hour, and then specific band of antibody reaction were obtained by chemiluminescence using horseradish peroxide substrate, milk, as shown in next slide, Fig 171 and 72. For quantification, ImageJ program was used, and for statistical analysis, the free software R was used. This is the result. So the abbreviation of PEL is the lysosome inhibitor, so peptide E64D and roipepsin, which adjusted to the final concentration of 10 micromole. And CT is P301 L tau only, and CAC is a tau plus CAC. CAC plus PEL is a tau plus CAC plus PEL. The soluble is a soluble fraction, and ins is the insoluble fraction. So tau protein aggregation was reduced by CAC717 treatment and was not affected by the inhibition of lysosomal proteolytic enzymes like PEL plus PEL. Okay. The second cell level verification is using the messenger RNA. So verification of tau aggregation inhibition by CAC717 at the messenger RNA level. It is a bit complicated. Even after 24 treatment of CAC717 in N2A cells, I astonished no change was observed in the messenger RNA expression pattern analysis. So Fig 8.1 at the left side. String ties CTAB data processed with ball gone. Therefore, I evaluated the effect of CAC717 on the messenger RNA expression patterns of N2 cells expressing mutant tau. Since apoptosis induced by stress of the tau aggregation is expected in untreated cells, so NT, I expect that the messenger RNA expression pattern in the case of the CAC717 treatment would completely differ from that of the untreated tau aggregated L2A cells. Then it speculated the result. In normal N2A cells treated with CAC717 is no change. But plasmid tau was expressed, the pattern of the messenger RNA is a bit shifted as red colors. But with CAC treatment, the activation is normal response. But in the case of the aggregation of the tau protein, the cell change to the apoptosis as so the big entropies as infinitive in the data of the cells. So the right side is the models of the shift of the messenger RNA pattern in the center of the normal, and activation occurs as expression of the tau protein. But in aggregation of the taus, would result in the apoptosis as a black spot. As expected, the messenger RNA expression pattern of normal N2A cells and also the CAC-treated N2A cells, NT, is blue groups. Mutant tau overexpressing no treatment group is a tau aggregation is a black. And tau overexpressing CAC-treated group red, so tau CAC, but distinctly different as expected. So it shows the tau aggregation was blocked by, or dissociation is induced by CAC770 treatment. This is done by the multivariate analysis as principal component analysis. The third level is the biological levels. So genetically engineered Drosophila was used for prepare for the AD models. The material method, the feeding and the layering condition and the drug administration method. The diet were algae, prepared according to the standard recipe for Drosophila experiment. CAC770 solution, concentration of about 2.5 milligram per milliliter, was stored at 4 centigrade in the dark and was added at 5% volume to the agar diet. Layering jar, 22 millimeter diameter times 69 millimeter height were used for layering at 18 centigrade before test and shifted to 29 centigrade to promote TS didn't after start of the adult test. Light illumination was 12 hours, 9 a.m. to 9 p.m. So main strains of Drosophila used. 3 strains of Drosophila were used in this experiment. The F1 individuals were kept at the layering temperature of 29 centigrade to synchronously express the transgene. The strain 1 is Oregon R is a wild type Drosophila. Strain 2, WUAS Tau R, so leading 406 double tryptophan. Tau models transgenic for mutant Tau, Tau R406W, which result in frontotemporal dementia in human. Strain 3, YW, GMR GAL4, RH1GFP, TAB GAL ATTS, rhodopsin RH1 and fluorescent protein GFP conjugated protein under the control of the photoreceptor neuron-specific promoter GMR and the TS promoter TAB GAL ATTS for neuronal cell evaluation. These 3 strains was used for the experiment. The first result, the extended lifespan of a mutant tau expressing individual in CAC717 treatment. Survival checks were carried out at least once every 3 days after start of the test, and the numbers of deaths was recorded until all the animals had died. Survival curves were generated using the Kaplan-Meier method, and statistical tests were performed using the log-log test in the analysis software R. The Tau R406 double expressing population, Tau, was reduced by half within 3 to 4 weeks after induction and died within about 5 weeks, while CAC717, 5% volume per volume treated group, Tau CAC, showed a significantly longer lifespan compared to Tau groups. Probability is 0.0001. However, the Tau non-expressing GFP or GFP-CAC group were longer, showed in effect 9. The second test is a climbing assay. Each measurement group, so 80 individuals, was placed in an empty layering jar and allowed to rest for 15 minutes before the bottom of the jar was gently tapped against the leaden part, causing all individuals to fall to the bottom. The number of the individuals that climbed at least 5 centimeters in 10 seconds was then measured. The mutant Tau R406W expressing population, so the Tau group, showed a marked decrease in climbing rate, which reduced by half within 2 to 3 weeks after induction. On the other hand, in the CAC717 treated groups, Tau plus CAC The decline in climbing rate was suppressed. Even 20 days after incubation, there was no difference as Fig 10 compared to the group without tau treatment, GFP or GFP-CAC. Okay. The last in vivo test is the effect of CAC770 treatment on rod cell degeneration in mutant tau expressing population. The photoreceptor R cells at 7 days after induction of mutant tau R406W were examined. In the standard diet group, many eyes shows a degeneration of R cells showed as arrow. A decrease in cell number and abnormal arrangement was shown in AC. On the other hand, in the CAC717 administrated group, many eyes shows normal number and normal arrangement of R cells, D to F. It was found that the damage of R cells due to mutant tau expression and aggregation was observed 7 days after induction, and that the effect of CAC770 in suppressing this damage could be evaluated. 11. The statistical analysis was shown in right side figures. The probability is less than 0.01. It's the last slide. The Western blot analysis of the brain of CAC717 treated Alzheimer's disease model Drosophila, the tau expression. The Drosophila had each group n equals 30 from human mutant tau expressing group TCT were dissolved in HBST 1% triton X100, 150 millimole sodium chloride, 10 millimole herpes, pH 7.4. And the soluble fraction and insoluble fraction were obtained. Western blot analysis showed that the amount of aggregated tau was reduced in the CAC770 treated groups as CAC compared to the untreated control group CT as shown in Fig 12. Okay. Oh, this is the last slide. Acknowledgement. I'd like to express my deepest gratitude to the members of the Faculty of Veterinary Medicine, Okayama University of Science, Professor Nobuyuki Kimura, and DVM Hayato Sato, and Professor Akikazu Sakudo and DVM Nodoka Iwatani, and Associate Professor Kohei Saeki. Thank you for your attention.